Okay, so we're in chapter 4, how neurons transmit information. Okay, a few reminders and announcements before we get going. Homework 4 is of course due this Sunday at 11 p.m. on La Lima as usual. And there's a change to the syllabus. So exam 1 was supposed to be, initially supposed to be this week, and it would be covering chapters 1 through 4. I'm going to open it next week so that people have time to get any questions answered about chapter 4. So the exam will be available starting on the 20th, which is a Monday, and will be open until the 26th. It'll be due at 11 p.m. on the 26th, and as noted on the syllabus, you're going to have only 1 hour to complete the exam, but it can be taken during any 1 hour period from Monday through Sunday of the week after this upcoming week. Okay, so starting on the lecture material, here in section 4.1, electrical activity in the nervous system. A little better background on electricity in the nervous system. Stephen Gray, who's considered kind of the father of electricity starting in the late 1600s, demonstrated that electricity could pass through the human body without injuring the individual, and he was famous for this demonstration where he would suspend a kid by insulated cords, so sometimes like silk cords, and create static electricity near the boy's feet, and show that foil and little bits of paper were attracted to the boy's face. So the static electricity passed through the boy's body to attract these bits of paper and foil near the face. A bit later, Luigi Galvani showed that electrical stimulation created muscle contraction in frogs' legs, and one of his experiments involved stringing up these frogs' legs on his, I think, the ceiling of his building during a lightning storm, and letting the electricity in the air stimulate the frogs' legs to move. A little bit after this, Fritsch and Hitzig showed that electrical stimulation of the neocortex can cause movement, and they showed this in animals. And after that, Bartholow showed that the brain of a conscious human can be stimulated to produce involuntary movement. He showed this during surgery of a live person. And then Caton came up with the voltmeter, which is a device that measures the flow and the strength of electrical voltages by recording electrical potential differences. So it's kind of a very early and basic level device for measuring electricity. And he used this voltmeter to measure electrical flow in the brain, so directly showing that there was electrical flow happening in the brain. And this laid the groundwork for the modern encephalogram, or EEG, which you might recognize. It's this way of measuring brain waves where you have these sensors typically embedded in a cap that are connected all over the head and measure neuronal activity underneath, and you get these nice recordings, these kind of like wiggly recordings. It's one way to measure like seizures or really basic broad processes. Just a side note, in case you're interested, there are actually video games that you can play these days using EEG. So there's some different types of brain waves that you can control consciously. One example would be called alpha waves, and we'll talk about this later when we talk about sleep and consciousness. But alpha waves can be controlled by relaxing or concentrating. And so there's some really basic EEG caps or sensors that you can buy, and they're connected to brains. So there's this game you can buy on Amazon, actually, where you control a levitating ball with your alpha waves. And then some labs, this is not available for commercial use, but some labs, I think, in Barcelona have created like system where you can play World of Warcraft and two-player Pong and things that are a little more complex using EEG signals. And this is called brain-computer interface, when you use signals from the brain to control a computer. It's an area of research that's becoming more and more popular, and they're coming up with more and more ways to use it. So another way that people are trying to use EEG is by creating music through brainwaves. So if you're interested in music or music therapy, this could be interesting to you. There's one example of scientists teaching people who can't move to use their brain waves to create drum beats. And it's a difficult process, but they're still kind of in the development for it. There's another example of people who can control the tempo, dynamics, and articulation of existing music through EEG. And then this is I hadn't heard of this actually before I was looking these things up for this lecture, but apparently there's these cat ears that you can buy that also rely on maybe alpha waves to control the orientation of these cat ears. Okay, so back to the course material. Helmholtz discovered or hypothesized, I suppose, that the signals in neurons are too slow to be just using electricity. And a bit later, Bernstein figured out that it's a wave of charge that passes from neuron to neuron rather than actual ions passing through from neuron to neuron. And he discovered that chemical signaling allows messages to travel along the neuronal axon and pass the message to the next neuron. Some ways that we can measure activity of individual neurons include inserting a microelectrode, which is a very, very small microscopic insulated wire or a tube filled with like salty water whose uninsulated tip is used to stimulate a record from those neurons. And an oscilloscope is a device that records changes in voltage over time. It's like a very sensitive version of a voltmeter, and EEG is like a bunch of oscilloscopes that record different that record the activity at a bunch of different sensors. Okay, and then Hodgkin and Huxley were very famous scientists who studied the axons of giant squid. Giant squids have these very large axons that make it easier to study them because they're so large. And they use these giant squid axons to understand electrical activity within the neuron. They received a Nobel Prize for this in 1939. And they identified the nerve impulse, which is this change in ion concentration across the cell membrane that underlies signaling between cells. They found out that the electrical activity in a neuron is caused by movement of intra and extracellular ions carrying positive and negative charges across the cell membrane, and that changes in the concentration gradient of these ions creates changes in the voltage gradient. And this is what underlies this cell signaling, and so that's what we're going to talk about for pretty much the rest of this chapter is this basic process. Okay, so section 4.2, the electrical activity of the membrane. One of the basic concepts to understand is resting potential. So this is when the neuron is not sending a signal, it's at rest. And resting potential is, in human neurons, is when the difference between the outside and the inside charge of the cell membrane is negative 70 millivolts. This is kind of the charge that the cell prefers. So the inside of the cell is negative 70 millivolts compared to the outside of the cell. And this is called stored potential energy. The negative charge is maintained on purpose by the channels, gates, and pumps in the cell membrane. And if you like, there's an animation on YouTube that you can that gives a little more detail and some animations. So there's 4 main particles that you need to know about that create the resting potential. There are these large protein molecules that mostly stay inside the cell, and those are symbolized with this A minus. And then there's potassium, K plus, chlorine, which we're going to show as Cl minus, and sodium, Na plus. There's a greater amount of negatively charged ions and proteins inside the cell, which creates that resting potential of negative 70 millivolts. There's more sodium and chloride outside of the cell, higher concentrations outside the cell, and higher concentrations of potassium and those large proteins inside the cell at resting potential. As I mentioned before, because the membrane of the cell is relatively impermeable to large molecules, so large molecules can't really move inside and outside of the cell, those negatively charged proteins remain mostly inside the cell. And then ungated potassium chloride and chloride channels allow potassium chloride to pass freely, but the sodium channels are gated, so that keeps out positively charged sodium ions when the cell is at resting potential. And then there's also these sodium and potassium pumps that extrude sodium from the intracellular fluid and inject potassium into it. So those pumps kind of forcefully bring sodium in and potassium out. A graded potential is a small voltage fluctuation across the cell membrane. So small increases and decreases in the membrane potential of a cell compared to resting potential. If positive voltage is applied to a cell, the charge across the cell increases, it becomes less negative, and this is called depolarization, an increase in the electrical charge across the membrane. This is typically due to the inward flow of chlorine or sodium ions or the outward flow of potassium ions. In contrast, repolarization is when the charge of the cell becomes more negative again. An action potential is essentially the basis of a cell signal, of the signals of the cell, and this is a large, brief change in the potential of an axon membrane. So during an action potential, the membrane potential becomes much more positive, up to positive 30 millivolts. To cause this, a large amount of sodium quickly enters the cell. This causes the cell to depolarize. The membrane potential becomes very positive, as you can see in the figure on the right. The action potential is triggered when the membrane potential reaches negative 50 millivolts, and we'll kind of talk about how that threshold is reached in just a moment. After the sodium has quickly entered the cell, potassium then rushes out of the cell, which causes the membrane potential to again plunge to the negative values, and the membrane potential overcompensates just a little bit, and this is called hyperpolarization. So this little dip at the end of the action potential is hyperpolarization, and then it rather quickly returns to resting potential. There's some detailed explanations of action potentials if you want, or a more simplified explanation if it's still a bit confusing. Okay, so voltage-sensitive ion channels are a big part of the action potential as well. So there are those gated sodium and potassium channels decide whether those ions will be allowed to go in and out of the cell. When the action potential is happening, these channels open and close based on that threshold voltage of negative 50 millivolts. So if the membrane potential gets to negative 50, then the action potential is initiated. It's kind of an all or none. So if it's at negative 60, it won't trigger an action potential. It'll just go back down to negative 70 without hitting that peak. So negative 50, we'll say negative 50 for simplicity. Sometimes it's a little bit different in different types of neurons, but at negative 50 millivolts on average in human neurons, this threshold of excitation is reached, and that's the point at which an action potential will start. During that, both the sodium and potassium channels are triggered to open, but sodium channels are a little bit more sensitive, so they open more quickly. They open first, and that's what allows that quick influx of sodium and allows the membrane potential to become very positive, up to positive 30. When the membrane potential hits about positive 30, that's when the potassium channels have opened and potassium's rushing out of the cell, and that causes that dip down to repolarization, the slight overcompensation of hyperpolarization. The name, there's a name for this period after which an action potential cannot be triggered. It's called the refractory period, and this prevents, it's a really brief period, about 1 millisecond, that happens after the action potential where another one cannot be triggered. And it's during that hyperpolarization period. Okay, on this slide, there's just a showing what the potassium and sodium channels are doing during the action potential. So we have at resting state, this type of sodium channel is not allowing sodium in. This type of potassium channel is not allowing potassium out. Number 2, at depolarization, sodium is rushing in, causing this membrane potential to become positive and causing the action potential. Potassium is not quite rushing out of the cell yet. And this continues to happen in the rising phase of the action potential. And then in 4, the falling phase of the action potential, or repolarization, the sodium is no longer rushing into the cell and the potassium is rushing out of the cell, which is returning the cell membrane potential to the resting potential. And there's a little undershoot that's happening while potassium is still rushing out of the cell, and then we get back to membrane resting potential at negative 70 millivolts. So a nerve impulse is the propagation of an action potential along the membrane axon. So the way that a message is sent down the axon is one action potential after another is triggered. Depolarization at one part of the axon triggers depolarization at the next part of the axon, so that this action potential moves down toward the axon terminal. The refractory period of each successive part of the axon keeps the signal from traveling back toward the cell soma. So if you remember, there was that little dip that prevents another action potential happening immediately after one action potential, so that makes it so that the action potential propagation doesn't move back toward the soma. And there's a nice video of this if you like. So in human neurons, the myelinated axon, remember the myelin sheaths are these fatty coverings that the glial cells put over the axon to help the signal move more quickly. So action potentials can't occur where the myelin sheaths are covering the axon. There's just, they would be covering the receptors. So there are these little gaps between the myelin sheaths called the nodes of Ranvier. And these small areas are covered with voltage sensitive channels. So the channels that we were just talking about. And action potentials skip from node to node. So one action potential happens at one node of Ranvier, and that triggers the next one. And that's called saltatory conduction. Saltatory means like a leaping conduction. So you can imagine it leaping from one node to the other, sort of leaping over these little fatty sheaths. Why our axons are set up like this? It takes less energy to skip from node to node as compared to going straight along the axon. And myelin creates faster speed of conduction for the signal, as we've talked about before. So it's important for the myelin to be there, and the nodes of Ranvier are just a way to work around the myelin. So we talked about before about how multiple sclerosis, or MS, is an autoimmune disorder where the immune system attacks the myelin sheaths. You might be able to understand it in a little more detail now that we've covered some of a little bit more about the myelin sheaths and how signals are transferred through this axon. So just to review, MS is characterized by motor dysfunction, a loss of sensory function, depression, pain, fatigue, and people eventually do die from it. And in MS, the immune system attacks the myelin and damages it, which causes difficulty for individual neurons to signal from the brain. And this may, scientists think it may cause iron to build up because of reduced blood flow to the brain. And if you are interested in MS, there's a couple videos. This first one is more about the clinical characteristics of MS, and the second one is about people with MS describing their lives with this disease. Okay, and neurons use the EPSPs and IPSPs to determine whether or not an action potential is going to happen. And so if you look at this picture on the right, you can see that all these different, like the green and the blue, black, red, branches are axons of other neurons communicating with the dendrites of this cell. And 3 of these are EPSPs, so they are excitatory, and 1 is an IPSP, which is inhibitory. And the cell soma combines all of these and triggers an action potential if the sum of them are positive. So in the case of this particular example, there are 3 excitatory inputs and 1 inhibitory, so the action potential is going to be triggered. And this is triggered at the axon hillock, which, if you remember from earlier in the semester, is the place where the cell body and the axon meet. So it's triggered, this trigger zone or hillock is right at the beginning of the axon. The neuron has 2 different ways to decide which EPSPs and IPSPs are being added together. So there's all these signals coming into the neuron. How does it decide which ones they're grouping together to sum? And there are 2 types of summation. So there's temporal summation, where EPSPs and IPSPs that occur close in time are added together, and there's spatial summation. So EPSPs and IPSPs that are close in space, maybe on the same dendritic branch. Those are added together. So in temporal summation, you can see in terms of this EPSP, if they're widely spaced apart, they're not going to be added together. But if they're close in time, they're going to be added together to come a little bit closer to this -50 millivolt threshold, which, if you remember, is the threshold at which an axon is, at which a action potential is triggered. So maybe in this particular example, it looks like if there had been 1 more input, it would have made the cell, would have changed the membrane potential enough that an action potential was triggered. In spatial summation, EPSPs and IPSPs that occur close together in space are added together. So in the A part of this example, these 2 EPSPs are on opposite sides of the cell membrane and therefore are not added together. Whereas in the B part of this example, these 2 EPSPs are on the same side of the cell membrane. They're close together spatially, and so they are added together to form a larger EPSP. To initiate an action potential at this axon hillock, the total of the EPSPs and IPSPs must bring the membrane potential to that threshold of -50 millivolts. So in this example, there were these there's a little EPSP that wasn't enough to bring it to the threshold, followed by an IPSP, and then enough EPSPs were added together to reach this -50 millivolt threshold, which triggered an action potential. And for now, don't worry about section 4.4. I would prefer to cover this information in the next chapter, but if you're interested, go ahead and read that section.